Welcome to the deep dive. Let's talk about our immune system for a moment. It's truly this incredible, complex internal defense network, right?
Absolutely. A marvel of biological engineering, designed with countless components all working in concert, tirelessly protecting us from nearly everything.
But what happens when this intricate, multi-component guardian falters? We've explored autoimmunity before, where the system misidentifies self and attacks it.
Mm-hmm.
But today, we're diving into an even more critical scenario.
Mm-hmm.
When this mighty system simply fails to protect us from external threats that should be, well, easily neutralized.
That's the profound question at the heart of our deep dive today. We're stepping into the crucial, often challenging, but immensely illuminating world of immunodeficiency diseases.
Mm-hmm.
This isn't just an academic topic. It's an area of immunology that profoundly impacts millions of lives globally, highlighting just how fragile and vital our internal defenses truly are.
Our mission today is ambitious. To give you a mastery-level understanding of this field, like an elite professional. We'll unpack the fundamental distinctions between primary, which are inherited, and secondary or acquired immunodeficiencies.
Right.
We'll explore their incredibly diverse manifestations, delve into the sophisticated treatments that are truly transforming lives, and then embark on an extensive, comprehensive exploration of the most well-known acquired immunodeficiency, HIV/AIDS.
A huge topic in itself.
Exactly. We'll go deep into its intricate biology, its devastating global impact, the remarkable progress that's been made in treatment, and, perhaps most importantly, the immense ongoing challenges of vaccine development. Our goal is for you to walk away seeing the immune system through a completely new lens.
Get ready to discover some truly surprising facts, connect the dots in ways you might not have expected, and gain a profound appreciation for the immune system's intricate, delicate dance, and what happens when that rhythm is disrupted.
So when we talk about immunodeficiency at its very core, how do we define it, and why is studying these failures so incredibly important for immunologists?
Well, at its simplest, immunodeficiency is a critical failure of the immune system to protect its host you from disease-causing agents. Think of it as the shield being down.
Yep.
This is the opposite side of the coin from autoimmunity. In autoimmunity, the system mistakenly attacks its own body, its sense of self is lost. But with immunodeficiency, the problem is a failure of protection against outside invaders, bacteria, viruses, fungi, parasites.
And focusing on what goes wrong seems counterintuitive, but you're saying it's actually incredibly insightful. How so?
Precisely. By understanding where the breaks occur, which specific components are critical, and how they interact, we gain a much deeper appreciation for the complex processes of a properly functioning immune system. Each immunodeficiency, in a way, is nature's own experiment, starkly showing us the essential role of a particular gene, protein, or cellular process. When a critical component is missing or malfunctioning, its true importance becomes strikingly clear.
That makes incredible sense. With that core definition in mind, the very first fundamental distinction we need to make is between primary and secondary immunodeficiency. Let's start with primary immunodeficiency, or PID. What makes it primary?
Primary immunodeficiency, or PID, is an immunodeficiency that stems from an inherited genetic defect within the immune system itself.
Okay, inherited.
Because it's genetic, the defect is present at birth. And this often means that health problems start to manifest fairly early in life, sometimes even in infancy.
And it's not just one specific type of genetic glitch, right? It sounds like it can be incredibly varied.
Absolutely. The genetic defect can occur in virtually any gene involved in immune development or function. It could affect innate immunity, adaptive immunity, or impact either humoral, meaning antibody-mediated, or cell-mediated responses.
Right.
The severity can range enormously, from relatively minor conditions that might need little to no treatment, all the way to life-threatening disorders requiring major medical interventions.
And is this a rare phenomenon? Or more common than we might think?
Not as rare as some might think. In the United States alone, the estimated prevalence of primary immunodeficiency is about 1 in 500 individuals.
Wow, 1 in 500.
Yeah. What's also fascinating for immunologists is how these PIDs have served as invaluable nature's experiments. By studying these specific genetic defects, we've vastly increased our appreciation for how precise proteins and cellular processes are absolutely vital for the immune system to function correctly.
Right.
When a piece of the puzzle is missing, its true importance for the whole picture becomes strikingly clear.
So if that's primary, what then defines secondary or acquired immunodeficiency? How is that different?
Secondary or acquired immunodeficiency is distinct because it involves the loss of immune function that results from exposure to an external agent.
Ah,
This isn't about being born with a defect, but rather something you acquire during your lifetime that compromises your immune system.
and are there common examples of these external agents that lead to secondary immunodeficiency beyond just infections?
Yes, certainly. While infections are a major category and will delve deeply into one of the most significant, HIV, they're not the only cause.
Okay.
For instance, certain medical treatments, like immunosuppressive drugs given after organ transplantation to prevent rejection, intentionally induce a state of immunodeficiency.
Right, that makes sense.
Even severe social conditions, such as chronic malnutrition, are a huge global contributor.
And as you hinted, when we talk about secondary immunodeficiencies, one immediately comes to mind for most people.
Indeed. By far, the best-known secondary immunodeficiency is acquired immunodeficiency syndrome, or AIDS, which is the result of infection with the human immunodeficiency virus, HIV.
Right.
A key distinction from many PIDs is that secondary immunodeficiencies can often be reversed if the external condition causing them is removed. However, as we'll see, HIV/AIDS presents a much more complex and persistent challenge in that regard.
With that foundational understanding of primary versus secondary, let's really dive deep now into primary immunodeficiencies.
Yeah.
You mentioned there are hundreds of different genetic defects identified. That sounds like an incredibly vast and complex landscape to navigate.
It truly is. To date, more than 300 different genetic defects have been identified that cause these inherited immunodeficiencies.
300?
Yeah, categorizing them can be quite challenging because the immune system is so interconnected. We often loosely group them by whether they affect innate or adaptive immunity, or more specifically, B cells, T cells, combined B and T cells, phagocytes, or complement.
Ah.
However, a defect in one component can have ripple effects across multiple arms of the immune system. And surprisingly, different genetic defects can sometimes produce very similar clinical presentations, making strict categorization difficult.
So the consequences really depend on where that gene disruption hits, almost like a domino effect, wouldn't you say?
Exactly. The impact depends on the affected protein's specific role in the immune system, whether the mutation completely prevents protein expression or just interferes with its function, and how that ultimately impacts the overall immune response.
Right.
For instance, defects that interrupt very early hematopoietic cell development. The first steps of all blood cell formation can have widespread effects on everything downstream. Other genetic defects might more specifically block the production of certain lymphocyte populations, like B or T cells.
And when it comes to the adaptive immune system, you hinted earlier that T cell defects tend to have a much greater overall impact than, say, just B cell defects. Why is that?
Well, T cells play a pivotal, almost orchestrating role in directing immune responses. They're like the conductors of the immune orchestra, coordinating both humoral and cell-mediated responses.
Okay, the conductors. I like that analogy.
So defects that inhibit T cell development or function usually affect both antibody production and the cellular immunity needed to fight viruses, fungi, and intracellular bacteria. In contrast, if only B cells are affected, individuals primarily suffer from recurrent bacterial infections. But their T cell-mediated immunity against viruses and fungi often remains largely intact.
Given how varied and potentially severe these conditions can be, how are primary immunodeficiencies typically detected? What are the warning signs? And how has diagnosis evolved over time?
Traditionally, PIDs have often been detected early in life because parents bring their children to physicians with recurring severe illnesses and persistent health problems that just don't clear up.
Yeah, the kid who's always sick.
Exactly. Think of a baby constantly getting infections that a healthy child would easily fight off. There's a set of warning signs that physicians look for, often guided by organizations like the Jeffrey Model Foundation, which helps educate clinicians. Interestingly, early autoimmunity can also be a sign of an underlying immunodeficiency, reflecting a breakdown in the immune system's self-regulation.
But I can imagine diagnosis wouldn't always be straightforward, especially very young infants. Seems tricky.
You're absolutely right. Diagnosis can be delayed for several months, which is critical because early treatment is key for the most severe cases.
Why the delay?
Well, one reason is that infants are born with circulating maternal antibodies, passed through the placenta or breast milk, which initially provide a degree of protection and can mask early symptoms.
Ah, okay, so mom's antibodies hide the problem for a while.
Correct. To evaluate the possibility of immunodeficiency, physicians determine levels of antibodies and various immune cell types, monitor the infant's response to vaccines, and test for specific genetic variants, especially if there's a family history.
And this leads us to what sounds like a revolutionary step in early detection: newborn screening. Tell us about that.
Precisely. For the most serious PIDs, particularly those that block lymphocyte development, early detection is absolutely critical. A screening test was developed using standard heel or finger prick blood samples collected from newborns, the same samples used for other newborn screening.
Okay, the standard heel prick.
Yeah. It employs a rapid polymerase chain reaction, or PCR-based assay, to look for T-cell receptor excision circles, or TRECs.
TRECs. What exactly are those, and what do they tell us that's so vital?
So T-TRECs, or T-cell receptor excision circles, are like tiny molecular receipts left behind in the DNA of every newly formed T-cell.
Receipts, okay.
When a T-cell matures, it rearranges its genes to create a unique receptor, and this process creates these little circular bits of DNA that don't get used but are retained in the cell.
Ah, I see.
So if a newborn has very low or absent T-TRECs, it's a strong indicator that something is fundamentally wrong with how their T-cells are developing. Like, the T-cell factory isn't producing enough receipts because it's not making enough product.
That's clever.
Recommendations to screen every newborn for these defects were approved in 2010 and have been adopted by over 75% of babies born in the United States today.
That's fantastic uptake.
It is. This allows aggressive therapy to begin before live viral vaccines are administered, which could be life-threatening to an immunodeficient baby. For example, in California, this method detects immunodeficiencies in approximately 1 out of every 66,250 live births. It's a true game changer, giving these children a fighting chance from day one.
So we've talked about the spectrum of PIDs, and you mentioned the most severe forms. Let's delve into what's called combined immunodeficiencies, or CIDs.
Right.
What defines these, and why are they considered combined?
Okay, CIDs are among the most severe inherited immunodeficiencies because they result from either an absence of T cells or significantly impaired T cell function combined with some disruption of antibody responses.
Right, the T cell connection again.
Exactly. They're combined because T cells are typically crucial for robust B cell activation, the production of most types of antibodies, and processes like heavy chain class switching and affinity maturation. So if T cell health is compromised, humoral immunity or antibody responses will often be affected too. It's a double hit, essentially.
And what are the real-world consequences for individuals with CIDs? What does that mean for their health?
They experience increased susceptibility to almost all types of infectious agents. This is particularly true for viruses, protozoa, and fungi, but also for specific bacteria like Mycobacterium species, reflecting the importance of T cells in eliminating intracellular pathogens.
So a broad range of threats become dangerous.
Very broad. They also have reduced delayed type hypersensitivity responses and impaired cell-mediated cytotoxicity. Even viruses that are normally benign or live attenuated vaccines like measles and chickenpox can be life-threatening for these individuals.
And at the extreme end of the CID spectrum, we have severe combined immunodeficiency, or SCID. This is often what people think of when they hear bubble boy in popular culture, right? That term comes from this.
That's right. SCID represents the most extreme forms of CID, characterized by a complete or almost complete lack of functional T cells in the periphery. The underlying genetic defects often target very early steps in hematopoiesis, the lymphoid lineage, or the initial stages of T-cell development, and sometimes B-cell development as well.
So right at the beginning of immune cell production.
Exactly. What's crucial to grasp here is that SCID isn't one single disease, but a family of catastrophic immune failures, each hitting a different fundamental step in building a working immune system. It shows us just how many critical points there are where things can go wrong.
Can you walk us through a couple of the most illustrative genetic defect categories that lead to SCID? It sounds like there are several different fundamental pathways that can go wrong.
Certainly. One major category involves defects in the actual genetic rearrangement needed for T and B cell receptors. It's like our immune cells have a genetic puzzle, right?
Okay.
And they randomly combine pieces to make billions of unique receptors. But in these defects, due to mutations in critical genes like RAG1 or RAG2, the puzzle pieces can't be put together correctly.
So the unique receptors can't form.
Precisely. This results in a virtual absence of functioning T and B cells, though NK cells generally remain intact.
And the most common form of SCID, X-linked SCID, involves a different kind of signaling problem, correct? Not the rearrangement itself.
Yes, that's right. The most frequent culprit there is a defect in the gene encoding the common gamma, or vegan chain of the IL-2 receptor, located on the X chromosome, hence X-linked SCID or SCID-X1.
Okay, the common gamma chain.
This common gamma chain is also used by receptors for other crucial cytokines like IL-4, IL-7, IL-9, IL-15, and IL-21. So a defect here impedes signaling through all these receptors, fundamentally disrupting T-cell development and also impacting B-cell and NK cell responses.
Wow, one broken piece affects so many signals.
It does. This is the form of SCID that David Vetter, famously known as the Bubble Boy, had. Regardless of the specific genetic defect, the clinical picture of SCID is stark. Very low numbers of circulating lymphocytes and an inability to mount T-cell-mediated immune responses.
David Vetter's story is so impactful. He lived in a sterile isolation chamber for 12 years. That must have raised immense ethical questions, but also driven incredible research.
It did both. David's challenges profoundly impacted medical ethics, but his case also led to crucial research findings about SCID and significantly raised public awareness about immunodeficiency diseases. Today, such drastic isolation is mostly a temporary measure, only used until replacement therapies like bone marrow transplantation or gene therapy can be initiated. His challenges highlighted the immense complexities and risks, even with life-saving treatments, but ultimately spurred the scientific breakthroughs we see today.
So SCID is the extreme, but I've heard of something called leaky SCID. What does leaky imply here? Is it less severe?
Leaky refers to hypomorphic mutations in SCOD genes. These mutations only partially affect the expression or function of the proteins.
Okay, so some function gets through.
Exactly. This means you don't have a complete absence of T cells, but rather partially reduced numbers and impaired T cell and antibody responses. In some cases, there are even generalized effects on DNA repair processes.
Interesting.
A notable example is Omen syndrome, a version of leaky SCID characterized by the presence of oligoclonal autoreactive T cells, leading to chronic inflammation of the skin, swollen lymphoid tissues, and high levels of eosinophils and IgE antibodies.
Wow, so autoimmunity again.
It's a striking example of how a defect causing immunodeficiency can also paradoxically lead to autoimmunity. The system is both weak and misdirected.
Moving beyond SCID, let's look at MHC defects, often called bare lymphocyte syndrome. How do these impact immunity, and why are they considered less severe than SCID?
Well, these defects are generally not as severe as SCID because they don't block the fundamental development of lymphocytes. Instead, they impact the critical presentation of antigens.
The presentation part.
Right. If you have MHC class I deficiency, caused by mutations that reduce or eliminate MHC class I molecules, it impairs the positive selection of CD8 plus T cells in the thymus. This leads to depressed cell-mediated immunity and heightened susceptibility to viral infections.
Okay.
Similarly, MHC class II deficiency involves defects in the genes that control MHC class II expression, reducing the numbers of CD4 plus T cells because they can't be properly selected. While these are general immune failures, they typically don't cause the complete collapse seen in SCID.
We've talked about defects in the T cells themselves, but what about the organ where T cells mature, the thymus? What happens when its development goes awry, as in DeGeorge syndrome?
DeGeorge syndrome typically results from various deletions on chromosome 22, often affecting a gene called TBX1, which is critical for embryonic development of the heart, facial structures, parathyroid, and crucially, the thymus.
So it's a developmental issue affecting multiple systems.
Exactly. Patients present with a varied set of symptoms, including immunodeficiency, often abnormal facial features, hypoparathyroidism, and congenital heart anomalies, which are frequently the most critical aspect.
And the immunodeficiency part.
It varies widely depending on how much thymic tissue develops. In complete DeGeorge syndrome, there's no thymic tissue at all, leading to severe T-cell depression. But in most cases of partial DeGeorge syndrome, some residual thymic tissue is present, allowing for some functional T cells, and treatments like antibiotics are often sufficient.
Let's move on to Wiskott-Aldrich syndrome, or WAS, and other T-cell signaling deficiencies. This sounds like another critical area where T cells are present but not functioning correctly.
Exactly. WAS is caused by an X-linked defect in the WASP gene, which encodes a cytoskeletal protein vital for assembling and reorganizing actin filaments in immune cells. Think of these filaments as the internal scaffolding and transport system of the cell.
Okay, the cell's internal structure.
Right. This scaffolding is critical for proper immune synapse formation. the crucial point of contact between immune cells, and for intracellular signaling. The clinical manifestations of WAS, which usually appear in the first year of life, can vary, but commonly include eczema, thrombocytopenia, meaning low and unusually small platelets leading to bleeding issues, and impaired cell-mediated and humoral immunity, particularly low IgM levels.
So a range of symptoms.
Yes, and patients frequently experience recurrent bacterial infections and can progress to autoimmunity or B-cell malignancy.
And how was treated? What are the options?
For milder cases, treatment targets the symptoms,
Mhm.
transfusions for bleeding, and passive antibodies or antibiotics for infections. However, severe cases often require hematopoietic stem cell transfer, which can be curative. Beyond WAS, other genetic mutations in proteins involved in T-cell signaling pathways, such as various protein kinases or components of the NF-key activation pathway, can also adversely affect T-cell development, activation, and effector functions. These defects can also paradoxically lead to autoimmunity due to immune dysregulation.
Now, let's turn to hyper-IgM syndrome, or HIM. The name suggests something about IgM levels, but what's the core defect that leads to this? It sounds specific.
Hyper-IgM syndrome results from an inherited deficiency in either CD40 ligand or CD40L, which is expressed on T cells, or in CD40, which is expressed on B cells and antigen-presenting cells.
CD40 ligand and CD40. Okay.
The critical point is that the normal interactions between CD40L on activated T helper cells and CD40 on B cells or APCs are impaired. This interaction is an absolutely crucial costimulatory signal required for B cells to be fully activated by T cells.
So without that specific signal, what are the downstream consequences for the B cells and antibody production? What breaks down?
Without this costimulatory signal, B cells cannot mount normal responses to T-dependent antigens. This means they can't form germinal centers, undergo somatic hypermutation to improve antibody affinity, or, most critically, perform heavy-chain class switching.
No class switching.
Exactly. As a result, patients cannot produce IgG, IgA, or IgE antibodies. However, their B-cell responses to T-independent antigens, which don't require T-cell help, are unaffected. This explains why they have normal or even abnormally high levels of IgM, giving the syndrome its name.
Got it. High IgM because the others can't be made.
Precisely. And because CD40, L-CD40 interactions are also needed for T cells to activate dendritic cell maturation, this defect also leads to diminished cell-mediated immunity and increased susceptibility to intracellular pathogens, classifying it as a combined immunodeficiency.
Let's shift to hyper-IgE syndrome, also known as job syndrome. This one sounds like it impacts multiple systems based on the characteristics.
It does. Hyper-IgE syndrome, or HIE, is a multi-system disorder characterized by recurring skin abscesses, frequent pneumonia, eczema, and, as the name suggests, very high levels of IgE.
High IgE, right?
Patients often also present with distinctive facial abnormalities and bone fragility. The most common cause is an autosomal dominant mutation in the STAT3 gene.
STAT3, what does that do?
Think of STAT3 as a vital internal messenger, relaying urgent signals from immune communicators like IL-6. Without it, your specialized Th17 cells, which are crucial for fighting certain infections, just can't mature properly.
Right, the Th17 cells.
This explains the susceptibility to fungal and extracellular bacterial infections like Candida albicans and Staphylococcus aureus. And while the exact mechanism for the elevated IgE levels remains unclear, it's fascinating that despite their very high IgE, individuals with HIE don't typically show abnormal allergic responses, possibly because STAT3 is also involved in the signaling for those reactions. Quite a puzzle.
Moving on to B-cell immunodeficiencies. These sound like they're primarily about a failure in antibody production, as opposed to the T-cell issues we've discussed. What are the general characteristics here?
That's generally correct. Patients with inherited B-cell defects are typically prone to recurrent bacterial infections. This is especially true for encapsulated bacteria like Staphylococcus, Streptococcus, and Pneumococcus, which heavily rely on antibodies for opsonization and clearance.
Encapsulated bacteria, right? Antibodies are key there.
Exactly. However, their immunity to most viral and fungal infections is usually normal because the T-cell branch of the immune system largely remains unaffected. The spectrum of these diseases is broad, ranging from a complete absence of mature B cells and immunoglobulins to a selective absence of only certain antibody classes.
Let's start with X-linked agammaglobulinemia, or Bruton's hypogammaglobulinemia. Sounds serious. What are its hallmarks?
This is characterized by extremely low immunoglobulin levels and virtually no peripheral B cells, less than 1% of normal.
Wow, almost none.
Babies born with XLA suffer from severe recurrent bacterial infections. The underlying cause is a defect in Bruton's tyrosine kinase, or a BTA, an enzyme essential for signal transduction through the B-cell receptor.
BTK.
Without functional BTA, B-cell development in the bone marrow is arrested at the pre-B stage, meaning they can't develop into mature B cells capable of producing antibodies.
So development stops early.
Yes. Thankfully, with modern antibiotics and regular replacement therapy in the form of passively administered antibodies, often called intravenous immunoglobulin or IBDG, this disease is quite manageable, allowing patients to lead relatively normal lives.
And what about common variable immunodeficiency disorders, or CVIDs? The name suggests they're a bit more variable and maybe complex.
They are indeed complex and diverse. CVIDs are characterized by recurrent infections due to immunodeficiency, a marked reduction in one or more antibody isotypes, and impaired B-cell responses to antigens, all without any other known cause.
Okay, reduced antibodies and responses.
These conditions can manifest in childhood or later in life, sometimes referred to as late-onset hypogammaglobulinemia. Respiratory tract bacterial infections are the most common symptom, and they are typically controlled by regular immunoglobulin administration. Most cases of CVA have undefined genetic causes, and most patients have normal B-cell numbers, suggesting the primary defect isn't usually in B-cell development itself, but rather in their function or activation. Still a bit of a mystery in many cases.
And the most common of all the isotype deficiencies, you mentioned prevalence earlier.
That would be selective IgA deficiency, affecting approximately 1 in 700 individuals, making it surprisingly common.
1 in 700, yeah.
People with this condition typically have significantly lowered IgA levels, but normal levels of other antibody isotypes. Many, up to 70%, are asymptomatic and live full lives.
So many don't even know they have it.
Possibly. If symptoms are present, they typically involve a greater susceptibility to infections of the respiratory, intestinal, and genitourinary tracts, which are the primary sites where IgA is secreted. The core defect seems to be an inability of IgA-expressing B cells to undergo normal differentiation into plasma cells.
So far, we've focused heavily on adaptive immunity, especially T and B cells. But what about the innate immune system? Can disruptions there also lead to immunodeficiencies? Seems like they must.
Absolutely. Defects in myeloid cells, like phagocytes, or in components of the complement activation pathways also cause significant immunodeficiencies.
Phagocytes and complement. Okay.
These often manifest as depressed numbers of phagocytic cells or critical defects in their function, leading to recurrent microbial infections of varying severity. The problems can occur at several stages of phagocytosis, from cell motility and adherence to engulfing organisms, and, critically, killing them intracellularly.
Let's start with leukocyte adhesion deficiency, or LAD. What's going on there? The name gives a clue.
LAD is rooted in a defect in the common beta chain, or CD18, of integrin family adhesion molecules. Think of these molecules as the sticky feet that leukocytes use to grab onto blood vessel walls and crawl out into infected tissues.
Sticky feet, I like that, so they can't get to the infection site.
Essentially, yes. With LAD, these sticky feet are dysfunctional, impairing the adhesion of leukocytes to vascular endothelium, which limits their recruitment to sites of inflammation. This leads to susceptibility to infections with both gram-positive and gram-negative bacteria, as well as various fungi. It can be quite severe, with some individuals unfortunately dying within a few years, while others can survive for decades.
What about chronic granulomatous disease, or CGD? That sounds like a prototype for a specific type of defect.
It is indeed the prototype of an immunodeficiency affecting phagocytic function. CGD is caused by a defect in the NADPH oxidase enzyme.
NADPH oxidase.
This enzyme is crucial because it allows phagocytes to generate superoxide radicals and other highly reactive oxygen and nitrogen species. Think of them as potent internal bleaches, which are essential for killing phagocytosed pathogens.
The killing mechanism itself is broken.
Exactly. Without it, the phagocytes can engulf microbes, but they can't effectively kill them. So these patients suffer from infections with bacterial and fungal pathogens, but also exhibit excessive inflammatory responses that lead to the formation of granulomas, which are small masses of inflamed tissue.
And Chiari-Higashi syndrome, or CHS, that sounds like a more widespread cellular problem based on the description.
CHS is a rare autosomal recessive disorder caused by mutations in the LYST gene, which encodes a protein crucial for transporting other proteins into lysosomes and controlling lysosome size, movement, and function. Lysosomes are the recycling and waste disposal centers of the cell.
Okay, lysosomal function.
The symptoms are quite varied. Recurrent bacterial infections, defects in blood clotting, oculocutaneous albinism, and neurologic dysfunction. In terms of immunity, phagocytes produce abnormally giant granules, which are a diagnostic hallmark, but they cannot kill engulfed pathogens effectively.
Uh-huh.
There were also defects in cytotoxic T lymphocyte and NK cell-mediated cytotoxicity because the LYST protein is involved in delivering their cytotoxic killing proteins. Without early antimicrobial therapy and bone marrow transplantation, patients often succumb to opportunistic infections.
Let's consider Mendelian susceptibility to mycobacterial diseases, or MSMDs. This highlights a critical pathway for certain types of infection, specifically mycobacteria.
Yes, MSMDs are a group of immunodeficiency disorders characterized by single-gene inheritance of susceptibility to mycobacterial diseases like tuberculosis and leprosy.
Okay.
Their discovery highlighted the critical connections between innate and adaptive immunity and the absolutely key role played by IFN gamma in fighting these intracellular pathogens.
IFN gamma, right.
Normally, a specific pathway involving IL-12 and IFN gamma is essential for macrophages and T cells to effectively kill these bacteria. When any of the 6 genes within this critical IFN gamma IL-12 pathway are defective, such as those encoding IL-12, IL-12 receptors, IFN gamma receptors, or STAT1, patients become highly susceptible to mycobacterial infections. It really pinpoints that specific pathway's importance.
We've covered cell-based defects, but what about complement deficiencies? You mentioned complement earlier. Are these common?
Complement deficiencies are relatively common, actually. They can manifest either as a generalized failure to activate complement, such as with C4 defects, or as failures of discrete pathways or functions.
And the consequences.
Most are associated with increased susceptibility to bacterial infections and/or immune complex diseases. For instance, deficiencies in early complement components like C1q, C1r, C2, or C4 are strongly associated with a high risk of systemic lupus erythematosus, or SLE, because the body struggles to clear immune complexes.
Right, the clearance issue.
And deficiencies in later components often lead to susceptibility to specific bacteria like Neisseria.
Next, NK cell deficiencies. We've seen how NK cells are affected in some SCIDs, but are there specific defects just for NK cells? And what are the consequences?
Yes. NK cells are crucial for innate responses, including producing powerful type I interferons and directly killing virus-infected and tumor cells. So mutations that interfere with their development and function can lead to immunodeficiency and susceptibility to viral infections and certain cancers.
Okay.
While many SCID-causing mutations can also block NK cell development, there are rare selective NK cell deficiencies. For example, mutations in the GATA2 transcription factor necessary for mature NK cells can cause recurring viral infections like varicella and cytomegalovirus pneumonia. Similarly, mutations in the MCM4 gene uniquely affect NK cells, linking these deficiencies to severe viral infections and certain cancers. It shows NK cells have their own unique requirements too.
This is truly a deep dive into the specific components. Now, let's explore a fascinating and somewhat paradoxical aspect. Immunodeficiency disorders that disrupt immune regulation, sometimes leading to autoimmunity. How can a deficiency result in an overactive immune response? That seems counterintuitive.
This is where it gets really interesting, isn't it? The core concept here is that if the immune system lacks the proper brakes or regulatory mechanisms, even a deficiency in those regulatory components can lead to an overactive immune response that starts attacking self-tissues.
So losing the brakes causes a crash.
That's a good way to put it. Normally, powerful mechanisms like thymic negative selection and the activity of regulatory T cells, or TREGs, prevent self-reactive lymphocytes from causing harm if those regulators are faulty.
The self-reactive cells run wild.
Precisely.
Can you give us a couple of specific examples of conditions where this paradox plays out?
Certainly. One example is autoimmune polyendocrinopathy and ectodermal dystrophy, often called APEST. This is caused by a defect in the autoimmune regulatory gene AIR.
AIR, okay.
The AIR protein is expressed in the thymus, where it's like a teacher showing developing T cells what self looks like so they don't attack it later. If AIR is depressed, autoreactive T cells escape the thymus and can then cause organ-specific autoimmunity, leading to issues with endocrine functions and chronic candidiasis.
So the teacher's missing,
Yeah.
and the T cells don't learn self-tolerance.
Exactly. Another example is immune dysregulation, polyendocrinopathy, enteropathy, X-linked, or IPEX syndrome. This is caused by a mutated FOXP3 gene.
FOXP3.
FOXP3 is essential for the development and function of regulatory T cells, or TREGs. These are the peacekeepers of the immune system, suppressing self-reactive T cells. Without them, these autoreactive T cells go unchecked, leading to a systemic autoimmune disease affecting the bowel, pancreas, thyroid, and skin. Sadly, it's often fatal in early childhood.
So it's not just these specific syndromes. Autoimmunity can pop up in other immunodeficiencies too, correct? It's a broader theme.
Yes. Autoimmunity accompanies many other immunodeficiency disorders due to various defects in immune responses and their regulation. For example, low lymphocyte numbers, or lymphopenia, can ironically lead to autoimmunity by disrupting the normal balance that keeps self-reactive cells in check.
Mm.
Even deficiencies in early complement components can be linked to conditions like systemic lupus erythematosus, or SLE, because the body struggles to clear immune complexes, which can then trigger autoimmune responses. It's a complex interplay.
Okay, this has been an incredibly detailed look at the nature and causes of primary immunodeficiencies. Now, let's turn our attention to treatment. What are the current approaches and future hopes for treating these complex conditions? Seems like a huge challenge.
It is a challenge, but while there are no surefire cures for all immunodeficiency disorders, there are several promising treatment possibilities beyond just managing infections with antimicrobial agents. The general strategy often revolves around replacement therapy, essentially, replacing what's missing or isn't working properly.
So, replacing what's missing. Let's start with protein replacement therapy. How does that work?
For disorders that impair antibody production, the classic and most common treatment is the administration of missing immunoglobulins. This is done through intravenous immunoglobulin, or IVIG, which is essentially pooled human gamma globulin.
IV, right.
It effectively protects against recurrent infections by maintaining sufficient levels of serum antibodies. For other protein deficiencies, like certain enzymes or cytokines, recombinant proteins produced in the lab can be administered. Excellent examples include recombinant adenosine deaminase for SCID and recombinant IFN gamma for chronic granulomatous disease.
And then there's cell replacement therapy, like bone marrow or hematopoietic stem cell transplantation. This sounds like the big one, especially for SCID, right? A more fundamental fix.
It is. Cell replacement, particularly hematopoietic stem cell or HSC transplantation, is currently the primary potential long-term cure for SCID patients. By transferring HSCs from an immunocompetent donor, the recipient can develop a functional immune system. Success rates are highest when transplantation occurs early, ideally within the first few months of life, before severe infections set in.
Timing is crucial then.
Absolutely. HLA matching with the donor is also absolutely critical, as is careful management of graft versus host disease, where donor T cells attack the recipient's tissues. Despite the challenges, it has been life-saving for many.
This brings us to what feels like the cutting edge, gene replacement therapy. How does this work, and what have the results been like? It sounds incredibly promising.
Gene therapy is a growing and exciting alternative, especially for single-gene defects like ADA deficiency or X-linked SCID. The mechanism involves isolating a patient's own HSCs and then using a harmless retroviral vector to insert a functional copy of the defective gene into the cell's chromosomes. These corrected cells are then reintroduced into the patient.
Using their own cells, corrected.
Yes. For 8S-CID, gene therapy has been remarkably successful, with most patients achieving significant, long-lasting reconstitution of their T and B cells and dramatic immune improvement, with no serious adverse reactions reported.
That's fantastic news for AD-SCI.
It is. However, for X-linked SCID, while initial trials showed immune function restoration, some patients unfortunately developed leukemia due to the gene inserting near oncogenes.
Oh no. That sounds like a significant setback, a serious complication.
It was, but the research is clearly not stopping there. Are there improvements being made? New approaches, absolutely. Newer lentivirus vectors, a different type of retrovirus, are showing very promising results for X-linked SCID, leading to effective reconstitution of B, T, and NK cells with no evidence of toxicity so far.
That's encouraging.
And the truly cutting-edge research now involves correcting defective genes in a patient's own HSCs using the revolutionary CRISPR-Cas9 gene editing technique, which has shown promise in preclinical studies. This technology holds immense potential for the future.
CRISPR-Cas9, yes. We hear a lot about that. That's incredible progress. To understand these diseases even better, scientists rely on animal models. These are often called nature's experiments in the lab. Can you highlight some of the most important ones and what they taught us?
These animal models have been absolutely crucial for understanding how the immune system works by revealing the precise effects of specific mutations. By comparing animals with and without these defects, scientists can tease out intricate details of normal immune processes.
Let's start with the nude mouse. That's a famous one, right?
The nude mouse, discovered in 1962, has a recessive genetic mutation that results in it being hairless and, crucially, having a vestigial thymus.
No thymus.
Right. This means it lacks T cells, leading to a profound deficiency in cell-mediated immunity. From a research perspective, they are incredibly useful because they can permanently tolerate grafts from other species, making them invaluable for transplantation studies and for growing human tumors to test new cancer treatments.
And then there are engineered models, like RAG knockout mice and the ultimate model, the RAG gamma chain knockout mouse. What makes them different?
Yes, RAG knockout mice are specifically engineered to lack the RA enzymes, which are essential for creating T and B cell receptors. This means both B and T cells are completely absent.
Okay, a cleaner knockout than the nude mouse in some ways.
Exactly. But the truly ultimate model for certain applications are RAG gamma chain knockout mice. These doubly mutant mice are totally deficient in B, T, and NK cells.
Wow, all three major lymphocyte types gone.
Correct. This makes them the ideal choice for generating chimeric mice reconstituted with a humanized immune system, often called SCID Hu mice. Human hematopoietic stem cells can differentiate within these mice to produce human immune cells.
Ah, and that's crucial for...
This is crucial for HIV research, as HIV does not infect mouse cells, providing an invaluable animal model to test therapeutic and prophylactic strategies against HIV.
We've covered primary immunodeficiencies in incredible depth. Now let's turn our attention to secondary or acquired immunodeficiencies. Beyond HIDs, which we'll get to in detail shortly, what are some of the other diverse causes of these acquired immune system failures?
Certainly. One secondary immunodeficiency that has been recognized for some time but still has an unknown precise cause is acquired hypogammaglobulinemia.
Go.
This condition typically manifests in young adults who have very low but still detectable levels of total immunoglobulin, often with initially normal T cell numbers and function. These patients experience recurrent infections, and the condition is generally treated with immunoglobulin therapy, allowing them to lead relatively normal lives.
What about immunodeficiency induced by medical treatments or other agents? That seems like a significant category.
It is agent-induced immunodeficiency. A prime example is the use of immunosuppressive drugs, such as corticosteroids, which are prescribed after organ transplantation or to manage autoimmune diseases. These drugs suppress the immune system to prevent rejection or dampen overactive responses.
Right. It's an intended side effect in that case.
Exactly. Another common cause is cytotoxic drugs or radiation treatments used in cancer therapy. These treatments frequently damage rapidly dividing cells, including immune system cells, leading to a temporary immunodeficiency. Patients undergoing such therapies require close monitoring and often need antibiotics or immunoglobulin to manage infections.
It also sounds like age itself can be a factor in acquired immunodeficiency. Is that right?
It absolutely is. Both extremes of age are natural factors that affect immune function. Very young individuals, particularly neonates and premature babies, are highly susceptible to infection. While their basic immune components are present, the full range of innate and adaptive immune functions takes time to mature.
Okay, they're very young. And the elderly.
Conversely, the elderly also experience an increasing risk of infection and malignancies. Generally, their cell-mediated immunity is depressed with reduced T-cell diversity and a diminished B-cell repertoire, making them less adaptable to new threats. It's a process called immunosenescence.
This next point is quite striking, given how much attention HIV/AIDS receives. The source says that severe malnutrition is the single most common cause of acquired immunodeficiency, even dwarfing the number of individuals affected by AIDS globally. That's a profound statement.
It truly is. The scale of impact from malnutrition is immense and often underestimated. Think about this for a moment. Something as fundamental as not getting enough food impacts more immune systems globally than even HIV/AIDS. That's a staggering reality, isn't it?
It really is. Hard to fathom.
It affects both innate and adaptive immunity. For instance, very low protein-calorie diets are associated with reduced T-cell numbers and functions. And micronutrient deficiencies, like insufficient zinc and vitamin C, contribute significantly to general immunodeficiency and increase susceptibility to opportunistic infections. Furthermore, vitamin D deficiency has been linked to inhibiting macrophages' ability to act against intracellular pathogens like Mycobacterium tuberculosis.
So it's a multifaceted problem hitting the immune system from multiple angles and a highly preventable one at that, theoretically.
Precisely. Severe malnourishment is one of the most preventable causes of poor immune function, and its effects are particularly deadly when it combines with chronic infections like HIV/AIDS, tuberculosis, cholera, or malaria, creating a vicious cycle of disease and vulnerability.
We've laid the groundwork for immunodeficiency. Now it's time to tackle the most well-known acquired immunodeficiency: human immunodeficiency virus, HIV, and acquired immunodeficiency syndrome, AIDS. Let's start with its devastating history and current global status. Where did this all begin?
It began with the first recognition in June 1981 when physicians noted clusters of unusual opportunistic infections like pneumocystis, jirovecii pneumonia, and Kaposi's sarcoma in specific groups.
Right, infections that healthy people just don't get.
Exactly. The common feature across all these cases was a marked deficiency in cell-mediated immune responses and a significant decrease in CD4 T helper cells. Since the early 1980s, AIDS has escalated to pandemic proportions, with tens of millions infected and dying globally. As of 2016, approximately 36.7 million people globally were living with HIV infection. Sub-Saharan Africa remains the most affected region.
Despite those stark numbers, there have been some remarkable positive trends recently. Correct, things are getting better in some respects.
Yes, thankfully. The rate of increase in people living with HIV has slowed, and crucially, the number of infected children has decreased, largely reflecting significant declines in mother-to-child transmission.
That's huge.
AIDS-related deaths have also been halved since their peak in 2005, and new infections annually have slowed. These gains are largely attributable to international agreements, particularly the United Nations Declaration of Commitment on HIV/AIDS signed in 2001, which paved the way for expanded prevention, treatment, and education programs globally. This led to a significant expansion of worldwide access to antiretroviral drugs.
So there's progress, but the challenge isn't over by any means.
Far from it. Despite the advances, the rate of decline in new cases has slowed, with about 5,000 new infections still occurring every day. It will require significantly expanded efforts from the international community to meet the stated goal of ending the pandemic by 2030.
Yeah.
HIV remains a dangerous pathogen primarily because it infects and kills CD4 T cells, leading to profound immunodeficiency, coupled with its high mutation rate and ability to establish latent infections.
Let's turn to the causative agent itself. The retrovirus, HIV-1, a master of deception, as you put it. What kind of virus is it, and what's unique about its genetic information?
HIV-1, discovered in the early 1980s, is a retrovirus. What makes retroviruses unique is how they carry their genetic information. It's in the form of RNA, not DNA.
RNA, okay.
Yeah.
The opposite of us.
Right. When the virus enters a cell, it uses a virally encoded enzyme called reverse transcriptase to essentially rewrite its RNA into DNA, the opposite of the usual biological flow.
Reverse transcription.
Exactly. This newly made DNA copy, called the provirus, then integrates into the host cell's chromosomes. From there, it can be replicated along with the cell's own DNA. It can either be actively transcribed to produce large numbers of new viral particles, or it can remain latent, unexpressed within the cell until an activation signal triggers its expression. This latency is a major part of its deceptive nature.
I understand there's also an HIV-2. How does it compare to HIV-1? Is it the same kind of threat?
HIV-2 was isolated some years after HIV-1. Its prevalence is mostly limited to West Africa, and crucially, the disease it causes progresses much more slowly, if at all.
Much slower.
Yes. In some regions, a significant percentage of the population may be persistently infected with HIV-2 and yet experience a nearly normal lifespan. Scientists hope that studying HIV-2's more benign cohabitation with humans can offer insights into combating HIV-1.
And where did HIV-1 and HIV-2 come from originally? How did they get into humans?
Both HIV-1 and HIV-2 evolved from strains of simian immunodeficiency virus, or SIV, found in non-human primates.
SIV.
It's believed that HIV-1 jumped species from African chimpanzees and gorillas to humans in Cameroon in the early 20th century, likely through the hunting and butchering of these animals for food, known as the bushmeat trade.
The cross-species jump.
Yes. This led to the spread of the group M HIV-1 virus, responsible for 99% of global HIV-1 infections, facilitated by colonial-era changes like the expansion of train routes and growing population centers. HIV-2 similarly arose from SIV transfer from sooty mangabe monkeys.
Given its specificity for human cells, how do scientists study HIV in the lab? especially if it doesn't replicate in standard animals. That sounds like a major hurdle.
That's a significant challenge. HIV doesn't replicate in common laboratory animals. While chimpanzees can be infected, they rarely develop AIDS, limiting their utility, plus there are ethical and cost issues.
Right.
This is where SCID mice, which we discussed earlier, are invaluable. These are SCID mice reconstituted with human lymphoid tissue, allowing them to be infected with HIV-1 and serve as a model for studying HIV-1 infection, particularly in the development of new drugs.
Let's delve into the intricate steps of HIV-1's structure and life cycle. How does this virus manage to infect target cells so effectively? What are the mechanics?
The HIV variant has a well-characterized structure. It has a lipid bilayer envelope derived from the host cell, embedded with viral proteins GP120 and GP41, which form NV spikes responsible for attachment.
GP120 and GP41, okay.
Inside, there are 2 copies of its RNA genome, surrounded by a core containing key viral enzymes, reverse transcriptase, protease, and integrase. HIV specifically infects cells that express the CD4 protein on their surface, primarily CD4 plus T cells, but also macrophages and dendritic cells.
So CD4 is the main receptor.
Yes. The infection process begins with the high-affinity binding of the viral GP120 protein to the CD4 molecule on the host cell. This binding triggers a conformational change, exposing a binding site for a co-receptor, either CCR5 or CXCR.
A co-receptor too.
Once the co-receptor is bound, it initiates the fusion of the viral envelope with the cell membrane, releasing the viral contents into the host cell's cytoplasm. It's a multi-step entry process.
Once inside, what are the next crucial steps in the viral life cycle? Especially regarding its hidden threat of latency, where does it go from there?
After entry, the viral reverse transcriptase copies the RNA genome into a double-stranded DNA. Then, the viral integrase enzyme inserts this newly made DNA, now called the HIV provirus, directly into the host cell's chromosomes.
Into our own DNA.
Yes. This is a critical step because the provirus can then remain dormant or latent within the host cell for long periods without producing any viral proteins. These latently infected cells form HIV reservoirs, which are difficult for the immune system to find and for antiretroviral drugs to impact, representing a significant obstacle to a cure.
The hidden enemy.
Exactly. However, if the infected cell becomes activated, the provirus can be transcribed, leading to the production of new viral proteins and RNA copies, which then assemble into new virions that bud from the cell surface, often leading to the death of the infected cell.
You mentioned that HIV variants can shift their preference for co-receptors between CCR5 and CXCR4. How do these R5 and X4 variants shape disease progression? Does it matter which one the virus uses?
This tropism shift is an important aspect of HIV infection. R5 variants use the CCR5 co-receptor and typically infect macrophages and effector memory CD4 plus T cells, which are abundant in mucosal epithelia. These are often responsible for the initial infection and early dissemination.
Okay, R5 is early.
Generally, yes. However, as the infection progresses, often later in the course of HIV infection, the virus mutates and X4 variants emerge. These use the CXCR4 co-receptor and preferentially infect naive and central memory CD4 plus T cells.
And X4 comes later.
Often, yes. The rise of X4 variants contributes significantly to the accelerated decline in CD4 plus T cell numbers characteristic of full-blown AIDS.
And there's a specific, rather famous, genetic mutation related to CCR5 that offers a lot of hope, isn't there? The co-receptor itself.
You're referring to the CCR5 deletion mutation. Individuals who are homozygous for this specific deletion do not express CCR5 on the surface of their cells.
No CCR5 at all.
Correct. This makes them nearly totally resistant to infection with R5 strains of HIV, which are the most commonly transmitted sexually. What's remarkable is that these individuals appear largely unperturbed by the loss of this chemokine receptor.
Wow, so they're naturally resistant.
To the most common strains, yes. This discovery has been a significant glimmer of hope and has led to new ideas for HIV elimination. The Berlin patient, Timothy Ray Brown, is a prime example of this. An HIV-infected patient who received a bone marrow transplant from a donor with the CCR5 deletion, he has been virus-free for over 10 years without needing any antiretroviral drugs. His case is a powerful catalyst for ongoing research into co-receptor blockade as a method for viral elimination.
Let's trace the typical course of HIV infection in an untreated individual. How does it gradually impair immune function and progress to AIDS? What does that timeline look like?
The progression to AIDS, in the absence of antiretroviral treatment, begins with an acute stage immediately after infection, often with flu-like symptoms. During this period, the viral load in blood and body fluids is very high. significantly elevating the risk of transmission.
High viral load early on.
Yes, anti-HIV antibodies typically appear during seroconversion, usually 1 to 3 months later. After this, HIV-specific cytotoxic T cells begin to control the infection, leading to a rebound of CD4 plus cells and a stabilization of viral load, known as the viral set point. A higher viral set point generally indicates a more rapid progression of the disease.
So after that initial phase, there's often a long asymptomatic period. What's happening during that time? It seems quiet, but is it?
This can be a very long period, with a gradual decline in CD4 plus T cells, but typically no outward symptoms. The immune responses are working to keep viral replication in check. However, a critical aspect of HIV's pathogenesis is its high mutation rate.
Right, the reverse transcriptase errors.
Exactly. Due to errors made by reverse transcriptase, the virus continuously mutates, producing viral escape variants that are no longer recognized by existing immune responses. Despite a stable viral load in the circulation, as many as 1 billion new virions are produced daily, continuously infecting and destroying additional T cells. This rapid evolution presents a formidable challenge for both the immune system and for vaccine development. It's anything but quiet biologically.
And eventually, if untreated, this leads to AIDS. What defines the onset of AIDS clinically?
The hallmark of AIDS progression is the onset of opportunistic infections. The Centers for Disease Control and Prevention defines AIDS by a reduction in CD4 plus T cell counts to below 200 cells per microliter, or by the presence of specific AIDS-defining conditions such as pneumocystis jirovecii pneumonia, Kaposi's sarcoma, or severe wasting syndrome.
The CD4 count is key.
It is. The onset of symptoms is usually signaled by a rising circulating HIV load and a concomitant drop in CD4 plus T cells. Untreated, the average time from virus acquisition to death is about 8 to 10 years.
That paints a grim picture. What are the key pathogenic events, the specific mechanisms that lead to this gradual decline and progression to AIDS? What's happening under the surface?
One of the most significant pathogenic events is the extensive damage that occurs in the lymph nodes and the gastrointestinal tract. While the plasma viral load might appear stable during the chronic phase, biopsy studies show high levels of infected cells and structural destruction in lymph nodes, often long before the plasma viral load visibly increases.
Damage of the lymph nodes, okay.
Crucially, studies have shown that the gut is a major site of HIV replication and CD4 plus T cell depletion, with dramatic drops in crucial gut T cells occurring very early after infection.
The gut. Why is the gut so important here?
Well, intestinal Th17 cells are a primary target for infection and destruction. These Th17 cells normally play a vital role in maintaining the integrity of the mucosal barrier and regulating immune responses to gut microbes.
Right.
Their destruction, combined with the disruption of the mucosal barrier, allows gut microbes and their products to translocate into the body, leading to rampant immune stimulation and chronic inflammation.
A leaky gut, essentially, causing inflammation.
Yes. This creates a deadly feedback loop. The inflammation generates more activated CD4 plus cells, which are then highly favored targets for HIV infection and replication, accelerating the T cell depletion. CD4 plus T cells die through multiple mechanisms, not just direct lysis, including a type of programmed cell death called pyroptosis triggered by abortive HIV infection and killing by other immune cells. This widespread immune dysfunction also leads to neurological problems like HIV-associated neurocognitive disorder, where infected brain cells produce neurotoxins.
This sounds incredibly bleak for untreated individuals. But thankfully, we have something called antiretroviral therapy, or ART, which has truly been a game changer. How has ART transformed HIV/AIDS?
ART has revolutionized the landscape of HIV/AIDS, transforming it from a rapidly fatal disease into a manageable, chronic condition. Infected individuals can now live essentially normal lifespans.
Manageable. That's the key word.
It is. The success of ART lies in its ability to inhibit HIV replication by targeting specific steps in the viral life cycle.
So it's not just a shotgun approach, but a highly targeted strategy against HIV's unique vulnerabilities. What are the main targets for these drugs? Where do they hit the virus?
Scientists have brilliantly designed drugs that target different crucial moments in the HIV life cycle. Think of it as hitting the virus at its entry point, preventing it from copying its genetic material, stopping it from integrating into our DNA, or even blocking it from assembling new viral particles.
Multiple points of attack.
Yes. For example, reverse transcriptase inhibitors block the virus from converting its RNA into DNA. Protease inhibitors stop the virus from processing its proteins correctly, and integrase inhibitors prevent the viral DNA DNA from integrating into the host cell's genome. There are also entry inhibitors and co-receptor antagonists.
The real power of RT comes from combining these drugs, right? It's not just a single medication, it's a cocktail.
That's the key. The standard of care involves combination drug therapy, often called RT regimens. Individuals typically take a cocktail of drugs that inhibit multiple steps in the HIV life cycle simultaneously.
Why is the combination so important?
Well, treatment is now recommended to begin immediately after HIV diagnosis. The primary rationale is to prevent the rapid production of drug-resistant mutants. HIV mutates so quickly that if only one drug were used, the virus would quickly evolve resistance.
Right.
But hitting multiple targets simultaneously, it's like trying to evolve resistance to 5 different locks at once. It's incredibly unlikely. These combination therapies are incredibly effective at lowering the plasma viral load, often to undetectable levels, significantly slowing immune system destruction, and dramatically improving patient health. The dramatic decrease in AIDS deaths in the U.S. is largely attributed to this advance.
Despite these incredible advances, a complete cure for HIV remains elusive. What are the main obstacles still standing in the way?
The biggest obstacles stem from 2 problematic properties of HIV. First, the persistence of latently infected cells, particularly CD4 T cells and macrophages. These cells harbor the HIV provirus but don't actively produce viral proteins, making them invisible to the immune system and unaffected by antiretroviral drugs. They act as reservoirs of infectious virus.
The reservoirs, yes, the hiding places.
Exactly. The second obstacle is anatomical sanctuaries. The virus can persist in certain sites, such as the brain, that are not readily penetrated by antiretroviral drugs or fully accessible to immune responses.
So hard to reach places, too.
Right. There's intense research into finding ways to reactivate these latent cells in a controlled manner, hoping that then the immune system or drugs could clear them. But that's tricky, too.
Beyond treating infected individuals, RT has also had a profound impact on preventing HIV transmission. This is a huge success story, particularly concerning mother-to-child transmission, isn't it?
It is indeed one of the greatest triumphs of art. Globally, the rate of infection for infants born to untreated HIV-positive mothers was about 37%. By 2015, that number had dropped by about 2/3, meaning an estimated 1.6 million infections were averted through ART.
1.6 million averted infections, wow.
In the U.S., mother-to-child transmission has declined by over 90% since the early 1990s. CDC recommendations include combination ART for HIV-positive pregnant women, C-sections when appropriate, and infant ART immediately after birth. HIV-positive mothers are advised not to breastfeed if formula is available. This has reduced the U.S. mother-to-child transmission incidence to 1% or less, a truly remarkable achievement.
Incredible. And this success is spreading globally.
Yes, this success is also being replicated in resource-limited settings, where the WHO now recommends lifelong ART for all pregnant and breastfeeding HIV-positive women, regardless of infection stage. This provides triple benefits: improving maternal health, preventing mother-to-child transmission, and preventing sexual partner transmission. Several countries have officially eliminated mother-to-child transmission.
ART also plays a role in preventing adult-to-adult transmission, right? Through treatment as prevention and pre-P.
Absolutely. ART significantly reduces transmission by lowering the viral load in infected individuals. Furthermore, pre-exposure prophylaxis, or pre-P, approved by the FDA in 2012, involves healthy HIV-negative individuals at high risk of infection taking a daily pill. Pre-D has been shown to reduce the risk of HIV infection by over 90% in people engaging in high-risk behaviors.
RT is an incredible success story, but the ultimate goal for halting the pandemic is a vaccine. After more than 35 years since HIV's discovery, why don't we have an effective HIV vaccine yet? What are the formidable challenges? It seems like the Holy Grail.
It is. This is one of the most pressing questions in immunology. The challenges posed by HIV-1 are truly unique and formidable. The most significant is the extreme viral heterogeneity and its rapid mutation rate.
The constant changing.
Exactly. HIV mutates constantly, even within a single infected individual, creating a moving target for both the immune system's natural response and for vaccine design. Natural immunity rarely prevents viral spread due to the rapid generation of viral escape mutants.
So the virus just keeps dodging the immune system.
Precisely. For a vaccine to be effective, it must induce broadly neutralizing antibodies, or bNAbs, that can react with most or all of these diverse viral subtypes and variants for long-term protection, and this is incredibly difficult to achieve.
Can you give us a quick overview of some of the major HIV-1 vaccine trials and what they've taught us, good or bad? What's the track record?
Well, early trials using purified viral proteins showed weak neutralizing antibody responses and no protection from infection. Disappointing.
Yeah.
Subsequent trials focused on eliciting cellular immunity using adenovirus vectors. These trials were unfortunately terminated prematurely because they resulted in a higher infection rate in the vaccinated group, especially among individuals with pre-existing immunity to adenovirus.
Higher infection rate. How could that happen?
It was theorized that the activated memory T cells in response to the vaccine actually became easily infectable targets for HIV. This was a major blow to the research community.
A huge setback.
But then came RV144, which offered some glimmer of hope. This trial showed a modest but promising 31% reduction in infection rates in the vaccine group. While not perfect, it was the first time a vaccine showed any protective effect. Building on this, new trials are underway.
Let's dive a bit deeper into the quest for these broadly neutralizing antibodies, or BNAbs, which sounds like the key for an effective vaccine. What makes them so special, and why are they so hard to induce?
Okay, so a small percentage of HIV-infected individuals do develop BNAbs, usually appearing late, 2 to 4 years post-infection. These BNAbs are remarkable because they can neutralize many HIV-1 variants.
Okay, so nature can make them sometimes.
Yes. Research has identified 6 vulnerable sites on the viral surface protein that these BNAbs recognize. But here's the challenge. Imagine the virus is hiding behind a thick, sugary shield. It's called a glycan shield. Most antibodies just can't get through.
The sugar shield.
But these special BNAbs have an unusually long and flexible arm. Think of it as a super stretchy probe that allows them to reach past that shield and grab a vital part of the virus that it can't afford to change.
Wow, so specialized tool.
And these BNAbs also require extensive training through prolonged multiple cycles of D-cell activation by the constantly evolving virus, which explains why they appear so late. They undergo massive amounts of somatic hypermutation.
That sounds like an incredibly complex process to replicate with a vaccine. How do scientists propose to stimulate this kind of evolution?
That's the challenge, how to stimulate such extensive training early on. One strategy involves isolating BNAbs from infected individuals, sequencing them, and then working backward to identify their ancestral forms.
A sort of reverse engineering them.
Exactly. Scientists then aim to design a sequential series of vaccine immunogens that, when administered, would essentially guide the immune system through the necessary evolutionary steps to produce these broadly neutralizing antibodies.
Guiding evolution. Fascinating.
In parallel, isolated BNAbs themselves can be used for passive immunization to prevent or treat HIV infection, with promising results in animal models and ongoing clinical trials.
So while we await a vaccine, the progress in RT and prevention remains vital. That's the message.
Absolutely. The continued development and availability of new and improved antiretroviral drugs and sustained education on prevention strategies are essential for reducing the global spread of HIV and managing the lives of those already infected.
Well, this has been an incredibly comprehensive and insightful deep dive. It truly highlights the immense importance of our immune system's intricate cells and molecules in protecting us from disease, and what happens when that protection falters.
Indeed. We've seen that primary immunodeficiency diseases, resulting from over 300 different inherited genetic defects, range from the severe SCID conditions affecting T and B cells and even entire hematopoietic lineages, to milder defects impacting single immunoglobulin classes or complement components.
Yeah, the range is huge.
The advancements in early newborn screening for SCID have been remarkable, allowing for prompt protection and life-saving therapies like bone marrow or HSC transplantation, with gene therapy emerging as a hugely promising approach. We also touched upon the fascinating paradox of autoimmunity arising from certain immunodeficiencies, where a lack of regulation leads to self-attack.
And then, the critical acquired immunodeficiencies, particularly HIV/AIDS. We explored HIV's problematic properties, how it targets and destroys CD4+ cells, which are crucial for its elimination, its continuous rapid mutation, and its ability to establish silent latent reservoirs in cells.
Yet, despite these challenges, the development of combination antiretroviral therapies has been revolutionary. It has transformed HIV into a manageable chronic disease, significantly prolonging lives and dramatically reducing transmission, especially from mother to child. The power of pre-P in preventing infection in high-risk individuals is another testament to scientific progress.
So what does this all mean for the future of immunology? Given the extraordinary challenges posed by HIV/AIDS and the ingenious ways science is fighting back, what new fundamental insights into the immune system's very design might we uncover next, perhaps even leading to broader applications for other diseases?
And how might the success in preventing mother-to-child transmission of HIV inspire new solutions for other complex global health crises? This deep dive truly illustrates that the more we understand what goes wrong, the more profound our appreciation becomes for the incredible, intricate dance that is the human immune system.
We encourage you to continue your own deep dive, perhaps by reflecting on how these complex systems interact within the human body. Thank you for joining us on the deep dive.